Hello, welcome everyone. Good evening, everyone. Hello, hi, good evening, everyone. Can I have some quick comments if you can hear my voice clearly? Hello, all my students. Please write in your chat box. Quick comments, please. Quick comments, everyone. If you can hear my voice. everyone. If you can hear. Okay. Okay, hello everyone. Good evening. Very good evening to all of you. This is Vaishali Kamdar. Hi, this is Vaishali from Spark. And today we will be discussing a very important topic, that is biomolecules. Biomolecules, it is important as it is important as biology point of view. So biomolecules you can consider in zoology also, in botany also. So this is a common topic. So you can't consider. So biomolecules can be present in plant system. It can be present in animal system also. So you can't consider it in either of the side. So it is common topic, and it is common topic between chemistry and biology also. There's one chapter in chemistry also, that is biomolecules. Okay. So in that you need to discuss about all the molecules or all the chemicals which are present in the biological body. So this is a common topic, and you will be getting questions in chemistry also related to biomolecules and biology also. But we are discussing as biological point of view today. So don't go anywhere. Quickly join the class. Those who have joined, ask your friends to join the class because it will be very useful and important discussion today. So 1 hour, just give your 1 hour to me, and your how many questions? Okay, how many questions are asked from this chapter that we will be discussing? And after 1 hour, I am sure that you will be securing your those marks. So before jumping into the actual biomolecules, let me tell you, please follow us on this website or Instagram, Telegram, Facebook, whatever you want. Follow us so that you will get update that which video or which live class is going to be on YouTube. You can also follow us on YouTube also. You like, share, and subscribe the channel. First, subscribe. What are you all waiting for? Just subscribe Spark Academy channel and YouTube, and then you will be having all the notification. Press the bell icon so you will get all the notification regarding all the lectures. Yeah, I know that today is not a biology. Actually, biology sessions will be on Thursday and Friday, as I have told you before. But today you'll be getting one benefit because I'm taking class today also. So from Thursday and Friday, or instead of Friday, I'm taking class today. So that is biomolecules. Discussion of biomolecules will be starting. Okay, so those who have joined the class, please write in the chat box so that I can call you if your doubts are clear. So write whatever is your doubt in the chat box. Are you all able to hear my voice? Everyone, come on, quick comments I want. See, I'll be giving some very important informations regarding NEET related to biomolecules today. And I'll be sharing few tips and hints and tricks to remember certain important points. So don't go anywhere. Stay tuned with me. 1 hour. It's 5 till 6 o'clock. So not exactly 1 hour. Just give me your 55 minutes. 55 minutes to me. Okay, done everyone. Very good, very good. So this is a botany chapter-wise weightage. In that we have discussed. I was showing these points in my previous videos also. So this is there in the cell and cell cycle you need. Cell and cell cycle you need. So that is cell structure and functional. In that one chapter is that is biomolecule. Most of the time, three questions are asked from this chapter. Three questions in NEET, so 2 to 3 questions in NEET, 3 questions into 4 marks. So gives you 12, 12 marks. How many marks? 12 marks. So 12 marks, your 12 marks will be going to secure after this 55 minutes. Okay. So 3 questions we can we very easy peasy type of chapter. You will be studying it in chemistry also, and here it is repeating. So why don't you secure? Why are you not securing your marks? So stay tuned with me, and those who have not joined, just call or chat your friends that ma'am is live now. Vaishali ma'am is live now. Please, all of you, those who have not attended or not joined the class, they can join it now and clear all your doubts. Okay. So before jumping into the biomolecules, I'd like to ask all of you, if you have not downloaded the app, go to the Play Store in your phone or in your tab and download the app, Spark Academy app. Install it, download the app, and you'll be getting great discounts on all our courses. Various different types of courses are going on. Different courses are going on, and you can select of your choice: long-term course, short-term course, and crash course. Whatever courses are available right now, you can join whichever is convenient to you. And numbers are flashing on your screen. Numbers are flashing on your screen. You can contact us for admission also. So what happens after downloading the app? You will be getting free handwritten notes, free study materials, previous year questions, so free PYQs and free mock tests. So all these things will be free. So offline download study material and free. See, you'll be getting offline downloads of the material also. So you don't need to use your Wi-Fi all the time, your data all the time. Those materials are offline available. Okay, and you can call us and contact us. You can follow us in on any of these things: Facebook, Instagram, Telegram. Okay, any of these things. Even in Telegram channel also we are sharing the materials, and you will be getting updates on Telegram also, that which will be your live class on YouTube, when it will be, so you can schedule, you can plan accordingly. So now biomolecules. So as you all know, biomolecules, that means molecules which are present in the biological body. So molecules which are present in the biological body. Bio, that means in the biological body. Okay, what are molecules? You all know that matter. Okay, you all know about matter, or anything surrounding you, or you itself, you are made up of matter. And matter, that means what? Anything which has space, which occupies space, and which has weight. So weight and space, they are known as matter. So right now it's not chemistry class, I know that. Okay, but basic thing we need to keep on revising. Okay, so these are metals. Matters are made up of molecules. Molecules are made up of elements. Okay, and elements are made up of atoms. Atoms are made up of subatomic particles. Sorry. So subatomic particles. So here we'll be discussing those molecules which are present in biological body. They can be organic, they can be inorganic. But most of the time we are considering in NCERT, as they have said, that all the organic compound which present in the living body, they are known as biomolecules. So organic, that means carbon containing compound. Carbon containing compound, right? Carbon containing compounds present in the biological body, they are known as biomolecules. There are two different types of biomolecules according to their size: biomacromolecules and biomicromolecules. If they are, if their molecular weight, if their molecular weight it is greater than 10, greater than 1000 Dalton, then they are said to be biomacromolecules. If they are less than 1000 Dalton, they are said to be biomicromolecules. So macro, that means big, micro, that means small. But exceptions are everywhere. Here one of the exception, that is lipid. Lipid. Lipid is lesser. It is having lesser. Why it is considered as micromolecule? Because its molecular weight is lesser than, or it is around 800 Dalton, which is lesser than 1000 Dalton. Okay, but sometimes they remain attached, or most of the time they remain attached to each other in a cluster form. That's why they are present in the acid insoluble fraction. Acid insoluble fraction. Now you will ask me that what is this acid insoluble fraction? What is this fraction termed as? See, I'll explain you that. That's why I have kept this slide empty. Wait a second. See, okay, for example, you have a container with you. Okay, you can say, for example, a beaker. A beaker. So you have a beaker with you. In that you have kept a funnel, and in that funnel you have kept a muslin cloth. I'm drawing that muslin cloth or a cloth. So you have kept a cloth here, and what you are doing here? First, you will be making slurry of a tissue. How you can make slurry out of a tissue? Just collect the tissue from any living body. It can be plant, it can be animal, and that tissue, you grind that tissue with, this trichloroacetic acid. With what? Trichloroacetic acid. So what you are doing here? You are collecting a tissue, then mixing it with trichloroacetic acid and grinding it, mixing it. And then this particular slurry, you want to filtrate it. You are filtering this slurry through a cloth. Okay, you are filtering this slurry through a cloth. What happens here? You will get something here and something as retentant here. Something as retentant here. Where? Here. Here this cloth will be containing whatever present up, that is known as retentant. And this is called filtrate. Okay, so filtrate is also known as acid soluble fraction. And retentant is known as insoluble fraction. That means acid insoluble fraction. Is it clear everyone till this point? Okay, for example, this tissue is containing a higher amount of proteins. Okay, so in during making slurry, if this protein is breaking down into amino acids, this amino acid you will get here. Amino acids you will get here in filtrate. So it is but obvious that all the micromolecules, this will contain biomicromolecules. Biomicromolecules like amino acids, okay, like glucose, or you say for example monosaccharides, okay, amino acid, monosaccharide, and nucleotides. Nucleotides. See, you will ask me that why I have not written lipid over here. As I have written it in biomicromolecule, I told you that it is having molecular weight lesser than 800 Dalton, so it is micromolecule. Still, I have not written in acid soluble fraction. Yes, because lipid will be present here. This is exception. Lipid will be present in retentant, in acid insoluble fraction. Though they are they are having lesser molecular weight than the 100 Dalton, 1000 Dalton, but they remain in a cluster form. Because of that, they remain present in the retentant part. They can't be filtrate out as a filtrate. Okay, so lipid will be there here, and along with lipid, what it will contain? All the macromolecules. So this retentant also contain biomacromolecules. Macromolecules, that means amino acid join together to form a polymer. So all the type of polymers, that means polymers. All the type of polymers, whether it is polysaccharide, whether it is polynucleotide, that means DNA or RNA, that is known as nucleic acid, or it can be protein. All these macromolecules, they are present in the retentant, or you can say acid insoluble fraction. Even lipids are also present in that. You getting my point? So now we will be discussing macromolecules as well as micromolecules. So first we will be starting with micromolecules. You all know that when micromolecules join with certain type of bonds, then they can form macromolecule. Okay, so now a question can be asked in NEET exam, that which type of, for example, a question is asked that in that they have given you one protein, okay, along with the name of see, I'll give you one example: lysine, then fructose, and say for example adenine, and one more example they have given, that is mm, insulin. So which of these four they are present? They are present in the filtrate, and which of these four they are present in the retentant? Okay, so this will be in retentant, or it will be there in acid insoluble fraction. Here, because it's a macromolecule, it is protein. But this is amino acid, this is monosaccharide, and this is nitrogenous base. These three will be there in acid soluble fraction. Okay, I hope it is clear. So now you can solve this type of questions. So which contains acid, which will be there in retentant, which will be there in the filtrate. Okay, now it is about amino acid. We are starting with we are starting with biomicromolecules. So biomicromolecules, they are of different types, like it may be amino acids. So you can see amino acid structure here. This is how it will look like. Amino acid structure will be having a carboxylic group. an amino group, and a hydrogen. These three are common. This is common structure. And what is different? That is R chain. Depending on the R chain, there will be different types of amino acid. If instead of R here it is H, then it is the simplest amino acid named as glycine. So glycine cannot form optical isomers. It is not optically active because here it is only H. So H, H, CH, COOH and NH2. So amino group and COOH, that is carboxylic group. So that's why see this is also having amphoteric nature, that means amphoteric nature, that means it can be acidic or it can be basic depending on the number of carboxylic acid present in it, number of amino group present in it. So amino acids can be of three types: acidic, basic and neutral. One second. Acidic, that means if it contains more than one carboxylic group, then it is having this group in higher number, then it will show acidic nature. If it is having more than one amino group in it, then it will be showing basic nature. It is known as basic amino acid. If it is having both the things in equal portion, equal COOH and amino, then it will be having neutral, it is neutral amino acid. Okay, so now one most important point here, it is Witter ionic form. See what you have learned in class 7th, that compounds which can give out hydrogen, for example, this is HCl. If you are making aqueous solution of it, then it can form H3O plus ion, that is hydronium ion. Okay, and which will also give you chlorine ion. So in aqueous solution, if the only positive ion present in the aqueous solution, that is hydronium ion, these type of compounds, they are said to be acids. Okay, so here, for example, this is your Zwitter ionic compound. So Zwitter ionic compound, I'll let you know what is actually it is. See here, right now it can give out this H plus and it can combine with the H2O to form H3O plus ion. So this is acidic right now. Okay, this is acidic right now. You can say it is acidic, and this is not giving H plus ion. It is not giving H plus ion. So this is acidic, and when it is irreversibly, see, reversibly, it can form this type of compound, this hydrogen here it is given to the amino group only. And if you look at this compound, positive ion is also here, negative charge is also there. Okay, positive plus 1 minus 1 gives you what is the net charge here? 0. So at which point it will give you 0 net charge. Net charge is 0 here, and this type of compound, this compound is known as Witter ionic compound, where the net charge of the compound is 0. Where the net charge of the compound is 0 is known as Witter ionic compound. Okay, so I hope you are getting this point. Okay, and pH, when you can get this net charge 0, that means pH of the Zwitter ionic compound, or pH at which you are getting this Zwitter ionic form, that is known as Pi, isoelectric point. It is known as isoelectric point. Okay, so I know that R group, if R group changes, amino acids, types of amino acid changes. Instead of H, if there is CH3, it is, say it is alanine. So different amino acid depending on the R chain. Okay, so you can say the chemical nature or properties of amino acid depend on the R chain. So it is not only depending on the R chain, it is also depending on the carboxylic group as well as amino group. So properties of amino acid, if you want to say, one second, properties of amino acid depends on which three groups you can say: R group, carboxylic group, and NH2 group. That is amino group. So properties of amino acid depends on these three groups. Are you getting this points? If you have any doubt, you can ask me in your in chat box. Okay. If you don't have any doubt, be prepared for the question answer session after 10 minutes, 10-15 minutes. Okay. So now we are starting lipids. So lipids are ester of fatty acids. You know ester bond. You might be knowing in chemistry. If it is like this, it is called ester bond. Okay. So here it is glycerol, and here it will be fatty acid. So like this type of structure, it is of lipid. So lipids are made up of fat or fatty acid plus alcohol. Which alcohol is normally used here? That is glycerol. So depending on the number of the glycerol present in the lipids, for example, it is only one glycerol present esterified with the fatty acid, then it is known as monoglyceride. If two glycerols are there, triglyceride. Three glycerols are there, so mono, diglyceride, and triglyceride, depending on the number of glycerol. Okay, so they are insoluble in water and soluble in number of known polar organic solvents like ether, benzene, and chloroform. Lipids form the colloidal complex and get dispersed uniformly in water in the form of minute droplets. So complex is known as emulsion. You all know about this emulsion. You have studied in digestion also. That bile salt present in the bile juice, it is used to emulsify lipids or to emulsify fats. Okay, what is emulsification? A bigger droplet of lipid, when it is broken down into smaller droplets or smaller particles, that is known as emulsification. Then there will be mixture uniformly. These droplets, they are uniformly dispersed in the water, and that is known as emulsion. So basic component of all the lipids are fatty acids. Basic component of all the lipids, they are fatty acids. So function of lipids or function of fats, they fat stored in the adipose tissue serves as the efficient source of energy. Adipose tissue serves as an efficient source of energy. Fat serves as a thermal insulator. In the subcutaneous tissue, see, you all know that those who are fatty, हाँ मैम भी फैटी है बोलना नहीं, हाँ या सो दैट मीन्स, सी यहाँ पे मोटी चमड़ी बोलते हैं ना अपन. सो मोटी चमड़ी, दैट मीन्स beneath the epidermal layer, subcutaneous layer, there is more adipose tissue present which stores the fat. So जिसके अंदर भी फैट ज्यादा है, they'll feel less cool. ओके, उनको ज्यादा गर्मी होती है और बहुत ज्यादा ठंडी है, तो उसमें इंसुलेटर का वर्क करती है. So insulator, that means thermal, like a thermal insulator, they'll work as a thermal insulator. So cholesterol and phospholipids are the important constituents of the cell membrane. What is phospholipid? One most important phospholipid that is made up of a glycerol, phosphoric acid, and nitrogenous base. Glycerol, phosphoric acid. So glycerol is esterified with phosphoric acid and nitrogenous base. This type of thing, it is known as phospholipid. And one of the most important phospholipid, it is known as lecithin. which is mainly present in the most of the cell, most of the important cell of the neurons and all. Okay, so lecithin. So lipids are of different type on the basis of which kind. See, they are of different types depending on their whether phosphoric acid present or not. So depending on that, phospholipids, glycolipids, sometimes they are glycosylated. That means carbohydrate group is also present. So they are known as glycolipids. So glycolipids, phospholipids, all these are different types of lipids. Now sugars. Another micromolecule, sugar. So you all know that sugar are sweet and water soluble carbohydrates. So you know that carbohydrates, sugar, for example, this is glucose or ribose. When we are talking about the micromolecule, it will be present in acid soluble fraction. Where it will be present? Acid soluble fraction. Okay, but when they will be in polyform, I mean that means polymer form, polysaccharide, they'll be present in acid insoluble fraction. So there are mainly two types of sugar we need to discuss, that is hexose sugar and pentose sugar. This is hexose sugar and this is pentose sugar. Hexose, that means having 1, 2, 3, 4, 5 and 6 carbons. 6 carbons in it. Oh, I know you will be not able to see this color in that white background. Wait a second. So 6 carbon. Here you can see 1, 2, 3, 4 and 5 carbons. So this is known as pentose sugar and this is called hexose. Okay, so if this sugars, see they, which type of bond will be there if they are joined together? So they can form disaccharide. It is known as glycosidic bond, glycosidic linkage. Okay, and glucose together, they'll form a polymer, polysaccharide you can call. Then about nitrogen bases. Next it is nitrogen bases. So they are carbon compound with heterocyclic ring. They are carbon compound with heterocyclic ring. See this type of ring structure you can see here. See in pyrimidine there will be only one ring structure and purine it will be having two ring structure. So there are two purines, adenine and guanine, and pyrimidine there are three, cytosine, thymine and uracil. Now you'll be noticing that what ma'am has written here, that is one key, important, very important key to remember. These are purines and these are pyrimidines. So you can see here cut, what I have written, cut pyrated because silver is pure. Cut, that means thymine, cut, that means cytosine, uracil and thymine. Cytosine, uracil and thymine. These three are pyrated, that means they are pyrimidine or pyrimidine you can say. Because silver, that means in chemistry how you are writing silver, it is Ag. Ag, that is adenine and guanine. Adenine and G matlab guanine. Ag is pure. Silver is always pure. Pure, that means it is purine. So I know this type of keys do not have any type of specific, you can say meaning. So you need to think logically here. We have made this, I have made this sentence just to remind, just you can remember these things easily. That cut is pyrimidine or it is pyrated one. Ag, that is silver, adenine and guanine, they are purines. Okay, so there are two types of nitrogenous base at pyrimidines and purines. Depending on the cyclic nature, they will be having one, not nature, cyclic structure, they'll be having two ring structure. They are known as purines, have a fused ring, and have a single heterocyclic ring. It is pyrimidine. Okay, so now see this new nitrogenous base. I'll explain you how this nitrogenous base. One second. How these nitrogenous base they are important. When this nitrogenous base, where's my pen? This nitrogenous base attached to the pentose sugar, this is called nucleoside. This is known as nucleoside. Okay, and when this is still attached with a phosphate or say phosphoric acid, then it is said to be as nucleotide. And what is DNA and RNA? All the nucleic acid, the nucleotides together join together with a phosphodiester bond, they'll form polynucleotide chain. And this polynucleotide chain, it is, you are saying it as, you are recognizing it as nucleic acid. Either it can be DNA or it can be RNA. So there are two types of nucleic acid, deoxyribonucleic acid and ribonucleic acid. So what is the difference here? In this pentose sugar, here one oxygen is removed. That is why it is known as deoxyribose sugar. Only that much difference between DNA and RNA. And one more difference, it is with the adenine, it is with the thymine and uracil. Okay, it is with the with the nitrogenous base, presence of nitrogenous base. ATGC, ATGC, AT, these four. I am writing it. A, T, G, and C. These four are present in DNA. Okay, these four are present in DNA. But instead of T, instead of thymine, there will be presence of uracil in case of RNA. And RNA is single-stranded, DNA will be double helical in nature. Okay, so double helical DNA, see Watson and Crick model will be discussing again. We will be discussing again in molecular basis of inheritance. So it will be like this. Two opposite direction anti-parallel chain of polynucleotide that will form DNA. DNA has two structure, left-handed and right-handed one. Okay, we'll be discussing it later on. Right now, we are moving towards the enzyme. That is last point of this chapter, enzymes. So enzymes, they are also known as biocatalyst. Biocatalyst, that means catalyst which is present in the biological body. So what is the role of catalyst normally? Catalyst which is not taking part in the reaction, but it can increase the rate of reaction. That is called a catalyst. So enzymes are those biocatalysts which can enhance the rate of reaction. Okay, they can speed it up and they can enhance the rate of the reaction. They are not actually taking part in the reaction itself. So you can see here one graph given here. One second. So as I have included this graph here, you can see here when activation energy, you can see activation energy with enzyme. So this is activation energy with enzyme. So you can reduce the amount of activation energy when enzyme is present. So this is activation energy you are requiring without enzyme. This is transition state. See what is transition state? See, normally what happens, enzyme reacts with substrate to form ES complex. And this ES complex will form EP, that is enzyme plus product, enzyme and product complex, and then there will be the formation of E plus P, that is enzyme and product. So substrate, here it is transition state. These are called transition state. Okay, at the transition state, you require an activation energy to convert the substrate into product. And for if you are having enzyme with you, this activation energy you can reduce. Okay, you can reduce amount of activation energy with enzymes. With enzyme. Enzymes are of two types. Excuse me. Enzymes are of two types, simple and conjugated. Simple enzymes, see, enzymes are mostly made up of proteins. They are mostly made up of proteins. But you can't say all proteins are enzyme. You can't say all proteins are enzyme. And sometimes they are made up of proteins plus some non-proteinic part is also there to make them activated enzyme. So this is how it will be, non-protein part plus the protein part. So non-protein part it is known as cofactor, and protein part it is known as apoenzyme. Okay, all together it will form holoenzyme. So they are of two types, simple and conjugated, and there are some factors which can influence enzyme activity, which can affect enzyme activity, that is temperature, pH, substrate concentration, and enzyme concentration. If you are increasing temperature, so at the low temperature they remain inactive. At the high temperature, they get denatured. So there is one optimum temperature for actually high, or you can say high productivity or high efficiency of enzymes. Okay, so at that temperature they work highest, their work or their efficiency is highest. Below that they decreases, above that also decreases. So optimum temperature, they can be more functional, more efficient. Like same in case of pH, at the low pH, they'll be not working. At the high pH, also they get denatured. So they are also pH sensitive. They work in the optimum pH only. Substrate concentration. See, if you keep on increasing substrate and enzymes are limited, so for certain period of time, enzyme activity increases up to certain extent. Then it becomes constant. Then it becomes constant because all the enzymes are occupied with the substrate. So there will be no free enzyme available to bind with the substrate if you are keep on increasing the substrate. So in that condition, you'll get this type of graph. You'll get this type of graph. So up to certain period it increases, then it become constant. Okay, so same type of thing happens with the enzyme concentration. If you are increasing enzyme concentration up to certain extent, it is increasing. Keep on increasing, keep on increasing. Then you are deficient for the substrate now. No substrate is available to bind with the enzyme, and you are keep on increasing the enzyme concentration. Then enzyme will bind with what? There is no substrate available. So in that case also it is becoming constant. Okay. So now types, or you can say classification and nomenclature of enzymes. So depending on their activity, there are 6 different classification or names of the enzyme. They are oxidoreductases. So they involved in the redox reaction. They can be also called as dehydrogenases. So oxidoreductases, that means in oxidation as well as reduction they are involved. Transferases, they just transfer one functional group from one substrate to the other. Okay, so transfer of the group, if they are involved in that activity, they are known as transferases. Hydrolases, that means they are using water molecules to break down one bigger molecule into two smaller molecules. So breaking down of the molecule with the help of water, if it is there, they are known as hydrolases. Okay, done. Very good. So now next it is lysis. So without involvement of water, you are breaking down one substrate into two, one molecule into two. So that is called lysis. I'm writing here. So this is with water, with addition of H2O, and this is without H2O. Without H2O, you are breaking down it. That is lysis. Then isomerases, you are converting it into the, see, structurally, if you write the formula, it will be same. CHO, C6H12O6, these numbers remain the same. But structurally, there will be differentiation. Structure will be changed, either optical or structural or functional change. So that type of thing, they are known as isomers of each other. Dextro and levo, D-glucose and L-glucose. So these are example of isomers. So when this type of structural variation they can create, they are known as isomerases. And next, last, it is ligases. Without this ligases, there will be no replication of DNA, no formation of DNA. You all know that DNA is synthesized or replication occurs in fragments form. And these fragments are ligated together. Ligated, that means ligation, that means they are also known as molecular glue. Glue, that means चिपका देना एक दूसरे को. So this चिपकाने का work if they are doing, then they are known as ligases. I hope this will help you out with your notes, and you can make notes out of this. And it will be very useful during your exam. I have covered all the points of this chapter. Now we are moving towards the question answer part. So before jumping into that, I am again reminding you, join or download the Spark Academy app, and you will get this link of the Spark store also. Without downloading it, you will be getting this store link in the description box. So that is Spark store, and you can buy or you can purchase anything from this: previous year solutions, full length test, study materials, mock test, crash courses are available, also long term courses are also available. So you can select or you can choose any one of that. Why you have all of you, please rejoin. Please rejoin, because I'm going to start the question answer part. Admissions are open offline as well as online. Offline is also working good. Long-term coaching is going on 10 to 4 with a lunch break of 15 minutes, and then regular coaching is going on 6 to 9 p.m. Okay, we have two branches, one at Narayanguda and one at Mehdipatnam. So you can join at anything which is convenient to you. Admissions are open for online coaching also. Online 2022 online coaching, and for JEE mains, for NEET, for EAMCET, you can select whatever is your convenience or whatever you prefer. So this is how on daily basis live sessions are going on. Even I am taking live sessions there, and you can, I mean, these videos are also recorded one. We are recording on daily basis. We are repeating all these things, and you will be getting all these videos afterwards also if you have downloaded the app. So it will be stored. So you can check it out afterwards if you can't attend at that time also. And on daily basis, we are giving assignments. We are checking those assignments also, and the test result we are considering. We are talking to the students why they are not getting average score or higher score. What is the problem? We are clearing doubts. We have a separate doubt clarification session every week. That's a plus point of Spark, that every week we are keeping one doubt clarification session and one revision session. So in the whole week, whatever thing is done, that can be revised in that week only. Okay. and the complete revision is done, but in that week also we are revising. Total revision, total doubt clarification is done. So this is our methodology, basic concept, shortcut methods, all model questions, exercise questions, PYQs, chapter wise tests, weekend test, grand test, and test analysis. Now first question. So these are PYQs. PYQs. A phosphoglycerate is always made up of what? A phosphoglycerate is always made up of what? Phosphoglycerate, it is made up of what? It is a saturated or unsaturated fatty acid esterified to the glycerol molecule with a phosphate group, is also attached. It's a type of phospholipid, you can say. So phosphoric acid or fatty acid, glycerol plus phosphate group, phospholipid. So then second question, so correct answer here, correct answer is B. Then essential chemical component of many coenzyme. Which of these is essential component of many coenzyme? So essential chemical component of many coenzyme, many enzymes are vitamins. So coenzymes are nicotinamide adenine dinucleotide or NADP contains a vitamin contain vitamin and niacin. Okay, so these are answers. This is the solution. With solution I am coming up. I have come up with the solution today. Okay, so that doesn't mean I'll be not giving you homework. Wait a second, I'll be giving that also. Now, transition state structure of the substrate formed during an enzymatic reaction is which type of transition state structure of the substrate formed during an enzymatic reaction? Permanent but unstable, transient and unstable, permanent and stable, transient but stable. Which of these is correct? In 2013, it was a transition state structure formed during the enzymatic reaction is transient and unstable. Then next, macromolecule chitin. Macromolecule chitin is fungal cellulose, is a nitrogen containing polysaccharide and heteropolymer of NAG. Heteropolymer of NAG. Next, the two polypeptides of human insulin are linked together by hydrogen bond, by covalent bond of insulin, phosphodiester bond or disulfide bridges. So it is insulin is a hormone consisting of its disulfide bridges. They are linked by disulfide bridges. Look at the solution part. Insulin is a hormone consisting of two polypeptide chain. Each chain is composed of specific sequence of amino acid residue. Okay. Post-translational modification result into the connection of the two chains by disulfide bridges. You can see here disulfide bridges. Okay. One second. The amino acid tryptophan is a precursor for synthesis of methionine and serotonin. Melatonin and serotonin. Melatonin and serotonin. Okay. Next, which of the following statement is correct with reference to the enzyme? With reference to enzyme, holoenzyme equal to apoenzyme plus coenzyme. That is correct. Holo equal to apo plus co. That means A is correct. Next, which of the following are polymeric? Proteins, lipids, polysaccharides. They are not polymeric. Proteins are polymer of amino acids. Polysaccharides are polymer of sugars. Nucleic acid polymer of polynucleotide, but lipids are not polymer. Most important point to note down here, lipids are not considered as polymers. Lipids are triester. They are the triester of fatty acid with the glycerol. So it is lipids which are not polymers. Then these are few homework questions. So I want your answer in a chat box, or after completion of class you can answer after the class also in comment section. So the catalytic efficiency of two different enzymes can be compared by the formation of product, pH optimal value, Km value, molecular size of enzymes. Next, which of the following is the simplest amino acid? I have included this point in this class only, so you'll get answer from that if you are concentrating on my words. Which of the following hydrolyzes internal phosphodiester bond in the polynucleotide chain? Next question, carbohydrate. The most abundant biomolecules on the earth are produced by which type of organisms? Which of the following statements regarding to protein is correct? So you can check out all the questions afterwards also and answer me in comment section. So again, I am reminding you, follow us. You can join the Telegram link which is given here, or it is there in the description box also. These are our results, EAMCET results. These are NEET results. Continuously, it is increasing. So we are growing continuously with our students, and all the faculty members are also working hard to clear their doubts, to practice them more. So join us. What are you waiting for? What are you all waiting for? Join us right now and contact us on the given number. Okay, this is our YouTube channel. Subscribe it, press the bell icon for the notification, like, share, and subscribe. All of you, please like, share, and subscribe. And thank you very much. Subscribe our channel to get notification, to get more information about related to this. Thank you so much. This is Vaishali Kamdar from Spark. Thank you very much. And thank you very much for everyone for listening me and for connecting with me. Thank you so much, all of you.